Hello, everybody, and welcome to the last part of Unit 2. This would be Unit 2.9 to 2.11. This was a really short and sweet little section, and for the most part on the FRQs, everybody did very, very, very well. So let's see what we got. 2.9 deals with the mechanism of transport, and here, once again, it's all about the membrane. We've talked a lot about that, and it's going to allow things in or out, which we've seen. And it's all about maintaining an internal environment that is different from its external environment. So that theme seems to be taking over this entire chapter, Unit 2. So some mechanisms of transport, and we kind of touched on them earlier in the chapter, but here's a quick review for those of you that maybe missed some things. We can use passive transport. Now, passive transport means no ATP, and it also means high to low concentration. And in the world of passive transport, it can either be simple, which would be your small, they don't necessarily have to be small, but the nonpolar portion is essential, and they can get right through easily. Active transport, in case you forgot, active transport is low to high, and here you need some sort of energy. And the energy can either come from, which we're going to find out in Unit 3, electrons, or it can come from ATP. So either electrons or ATP. That's your basic, keeping it simple, active transport. This is going to be polar molecules and or ions that need to go against the concentration gradient. This we'll see larger molecules as well, so things that you need to kind of get across with some help. Now, facilitated diffusion is going to come up in a second. That's, again, moving polar materials, but that's going with the concentration group, but let's just hang out with that. Endocytosis is endo, means to bring in, so it's going to move in large things, and it was going to kind of do it like this, where it pinches off a section of the plasma membrane, putting it into a vesicle. And that's sort of kind of how phagocytosis works in the world of white blood cells. Exocytosis is getting rid of large amounts of material. It's the exact same thing as you just did in endocytosis, except backwards. And then facilitated diffusion, be careful on that. And this is where many of you mess this up, and I don't want you to mess it up anymore. Facilitated diffusion is, again, high to low. It's diffusion, but it's facilitated, so it needs some sort of kind of like maybe channeled protein or something that will allow it to get through the cell membrane. So, for example, water needs what's called an aquaporin. So you may need help. But again, this is going along the concentration gradient. If you see those words, that means high to low or with it. So kind of like a little bit of a review, but a lot of people were getting all these words confused, and you can't do that because it's simple stuff that's going to cost you. So compartmentalization, this is 2.10. We only got to go up to 2.11. Here, once again, same deal, allowing internal and external environments to be different, but compartmentalization is your key of membrane-bound organelles. Why do you have endoplasmic reticulum, and why do you have vesicles, and why do you have lysosomes? You have all these things because you want to increase efficiency. You don't want these various different, working compartments to interact with each other in a negative way. You don't want them to interfere with each other. Everybody's got its own job, and if they all do it, and they do it well, and they do it quickly, the cell can get a tremendous amount of work done in a very small area. And keep in mind, your cells also have to work together, but this is within a cell. So compartmentalization is all about efficiency. Now, the origin comes from, of course, evolution, even though it's not going to be asked about supposedly on this test. It's the change in the genetic makeup of a population over time. Okay, so we know that. This here is very askable. The endosymbiotic theory. Endosymbiotic, endo means to bring in. Symbiotic means to work with each other. So in this case, we're bringing something in, and the things are going to work together. So what are we bringing in? there were these membrane-bound things that resembled mitochondria and resembled chloroplasts when they were these free-moving prokaryotic type, we'll call them cells at that point. They had their own ribosomes, so they could do protein synthesis. They had circular DNA. They had an inner membrane. They had all of this stuff. That's what mitochondria and chloroplasts have now, and that's what prokaryotes have. So it goes to say that there had to be some relationship between these mitochondria and chloroplasts. And also the key, many of you did say this, which was great, they have the ability to replicate within a cell without the cell actually replicating. Does that make sense? So if you had, let's say, 10 mitochondria in one cell, you can turn that into 20 mitochondria in that same cell without the cell having to divide. And if you look here, again, this is a long time ago. And what happened was, and again, over time, the infolding of the plasma membrane, okay? And in this case, this was an ancestral prokaryote, which again is going to move into, a little bit more of a, I guess we'll call it a sophisticated cell. And here you can see, in this case, the mitochondria being brought in. And in this diagram, you can see the chloroplasts being brought in. So in this case, this here was your ancestral photosynthetic, and it could be photosynthetic because it has chloroplast, or what was like a chloroplast back then. It's a photosynthetic prokaryote back then. And they were eukaryotic, so they relied on other food sources, if you will. And again, this took a lot of time. But that's the symbiotic relationship, the bringing in the endosymbiotic relationship. So let's see. Differences from prokaryotes. Prokaryotes lack membrane-bound organelles. So eukaryotes have a nucleus. Eukaryotes have endoplasmic reticulum, lysosomes. They have all of these membrane-bound Golgi apparatus, things like that. If you notice, prokaryotes do not have that. They have a nucleoid region where you will have your chromosome or your DNA. They also have these things called plasmids, which they do have the ability to actually kind of share with their neighbors. That's not what we're talking about here. So prokaryotic cells are different. They do not, no nucleus. They have genetic material, but they do not have a membrane-bound nucleus. They do have ribosomes for protein synthesis. They got that. Well, it's not on this picture, but it's probably these little specks right there. So those are your ribosomes. So prokaryotes can do business. If you look, there's their double membrane right there. So they got everything you need. That's how a eukaryote is different from a prokaryote. We have our videos, of course, endosymbiotic theory from Bozeman, some compartmentalization videos. And finally, the FRQ for this little mini section. Eukaryotes and prokaryotes survive on Earth today, and they're very successful. So, why, and everybody got this one right, which was awesome, why do eukaryotic cells compartmentalize? And they compartmentalize all about efficiency, so that everybody can be effective. And the jobs do not overlap, because remember, cells are simple, and what are they hoping to not have? Waste. No wasted time. And what's kind of cool is that in the world of chloroplasts, they produce some ATP, but that ATP goes right to glucose production in the Calvin cycle. Mitochondria, however, produce ATP for other cellular processes, for all the energy that's needed to do enzyme making, moving materials in and out. So the cell, again, is very, very, very efficient. Let's go back to look at question B. It is widely believed that some eukaryotic organelles evolved from a relationship with the prokaryotic cells. List 3 pieces of evidence, and many of you got 2 pieces easily, and some of you kind of slipped up. I gave you 4, actually. And let's see what we got here. We know that double membrane, that's 1. They have a circular DNA, that's 2. They have ribosomes, that's 3. And they do have the ability to reproduce. So prokaryotic cells can reproduce, but these guys can reproduce inside of a cell without the cell going through replication. So again, most of you got between an 8 and a 10 on this FRQ. So really nice job. It was a nice one. It was a short and it was sweet. So hopefully that's helpful, and I will see you in Unit 3.